Welcome to one of the past HSC exam question videos. In this video we're going to cover this exam question, which comes from the 2004 paper, and it's question 27 and it's worth 4 marks. In a second I'll read the actual question. Once I've read the question, you get about 5 seconds to pause the video. Once you've paused the video, attempt the question, and then press play when you're ready. And afterwards I'll go through the marking guidelines for all of these questions and show you sample answers in terms of what you could have written to get full marks as well. Right? So the question says, to study the effects of antibiotics on 3 strains of bacteria, A, B, C, agar plates were set up as shown. So a surface cover with bacterium A, surface cover with bacterium B, surface cover with bacterium C. Then we have this, and you need to read the legend here to get all the information. The plates were incubated at 37 degrees Celsius for 48 hours. The diagram on plates 1, 2, 3 depict the results. A. Write the conclusion to the experiment. B. Identify one safe work practice used to minimize risk associated with the handling or identifying microbes. And C. In humans, bacterial infections are often treated with antibiotics. Explain why the complete course of antibiotics should be taken, even if the symptoms of the infection have disappeared. So when you're ready, pause the video and attempt a question. Welcome back. Alright, so for the first one, A, it says write the conclusion to the experiment. So what was the actual outcome of the experiment? So to get 1 mark out of 1, all you have to do is write the correct conclusion. So in terms of the conclusion, we've got 3 bacteria, and you can see here this bacteria has completely grown. There is no, it's growth all over the place. This is meant to be, the white part is the clear zone. So in this case, bacteria A, quite a bit of the bacteria A has died. A bit of the bacteria B has died, whereas none of the bacteria C have died. So the actual antibiotic is most effective for bacteria A, somewhat effective for bacteria B, and not effective at all for bacteria C. That was basically the conclusion. So what I wrote is the antibiotics were most effective at killing bacteria A, somewhat effective at killing bacteria B, not effective at killing bacteria C. And all I had to do is look at the actual table to get that actual outcome. Alright, next one is identify one safe work practice used to minimize risk associated with the handling or identifying microbes. So in this case, we have to talk about risk factors, right? Marking guideline says to get your 1 mark out of 1, you have to identify one correct relevant safety practice minimizing risk associated with the handling or identifying microbes. So identifying microbes means actual experiment of doing it, and the handling part means after you've done the experiment, what do you do with the agar plates, for example, right? So one safety concern would be that the microbes we've grown might be dangerous. So to make sure that, so our safety precaution would be to remain those agar plates to remain sealed, right? We don't open them after we've closed them. We wash our hands after dealing with microbes in general, right? These could be 2 safety precautions. When it comes to handling microbes or identifying microbes, remember we have to actually burn the inoculating tube or sterilize it. So the problem is when we sterilize it on the Bunsen burner, we could burn ourselves. So that could be a safety concern. We could burn ourselves whilst trying to sterilize the inoculating tube. So the safety precaution would be just to take extra care, right? But these are 2 or 3 examples. You would have only had to mention 1, and you could have mentioned different ones as well, right? These are not the whole list. But basically the idea is my sample answer would have been like this. The bacteria might be harmful to humans, so to prevent the spread of the bacteria, the agar plates are to be kept sealed once it has been inoculated, right? Just a quick statement in terms of something to minimize the risk. If you wrote less, you can still get full marks. This is just says identify, that's it. And so far, we've got no marks here. Next question is, in humans, bacterial infections are often treated with antibiotics. Explain why the complete course of antibiotics should be taken, even if the symptoms of the infection have disappeared. And for this, we look at the next marking guideline. So it says to get full marks, so 2 out of 2, we need to have the identification of the need to minimize buildup of resistance by bacteria to antibiotics. Understand the need to maintain the effectiveness of antibiotics against the bacteria in the future. So why do we? What's the point of having basically bacteria not become resistant, and what does that what does it do for us in the future if we have them become resistant? That's what we have to talk about to get full marks. So in this case, we talk about antibiotics first and how they kill bacteria. And we say that if you don't take the full course of the bacteria, if you don't take the full course of the antibiotics, what that means is you kill basically almost all of the bacteria, except for the most resistant one. And because those most resistant ones survive, that means you're going to have antibiotic resistance developing, and that means you're going to have less successful treatments for the future in terms of patients that might come in. So to avoid all that happening, we would take our full course. That's what that basically means. Alright, so what I wrote for this one was the complete course of antibiotics has to be taken as even if most of the, I don't know what I wrote here, something random, but basically even if most of the bacteria have been killed, the ones that have survived will likely be the most drug resistant of the bacteria, right? So yeah, most might be dead, but maybe the ones who have survived might be the ones which are most drug resistant. Then I wrote, these drugs resistant bacteria can then divide and as a result produce a species of bacteria that is antibiotic resistant. Right? So because we have now these antibiotic resistant ones, they will be the ones that replicate and reproduce, and that means that we have a colony of resistant ones. This means that the future infections are not as easy to treat and new drugs have to be developed. So this is the outcome because of this antibiotic resistance. And then what I wrote at the end is, by finishing the course of antibiotics, the chance of development of antibiotic resistance is reduced, especially when it comes to obviously these bacteria, right? So talk about what antibiotics do, talk about what happens if you don't take the actual course, full course, and that gets you 2 marks out of 2. And where the question comes from, it came from these 2 dot points. So identify the role of antibiotics as in the management of infectious disease. That was obviously, oh, and there's also another one, discuss the implications of this one here. So discuss the implications of relating to antibiotic resistance and the role of antibiotics in the management of disease. That was number C. And number B and A came from the plan of first investigation to identify microbes in food or water. Right, that's where these questions came from. I hope that was useful.